Dear residents, today we will discuss the lecture on disease of wheat and their management. This is in the course of diseases of field and horticultural crops, which come in the sixth semester of the BSc AG program. So, in wheat, basically we encounter 5 or 6 diseases, that is foliar blight, then loose smut, then all the 3 rusts, then common or stinking smuts, then tundu ear disease. So we'll deal all one by one. So before dealing, since the courses of disease and their management, so before dealing with the disease, we should know some things. That is, what is control and management. So you all know that no plant pathogen has ever been wiped out from the face of the earth. So long as the pathogen survives and its host is cultivated, the chances of disease incidence will persist. So plant pathologists always aimed at the impossible task of eradicating the pathogens to control the disease. So nowadays the effort could be better called management practices, whereby the population of pathogen or the disease causing ability of the pathogen have been kept under check so that it is unable to cause noticeable damage to the crop. So you all know that the control is neither feasible nor possible. So only we can talk about the management. So in management we can bring the disease causing pathogen into check. So what should be the most important component of the IDM? The most important components of IDM are, one should use the healthy, pathogen-free or treated seeds. There should be avoidance of weeds, ratoons, stubbles of the cereal crops and overlapping generations. Then one should not plant the crop in sick soil. Then use of biological control should be promoted to reduce the soil-borne pathogen. Then adoption of cultural practices like intercropping, mulching, to minimize the disease buildup should be made. Then judicious inputs like water, fertilizer, organic matter and plant protection chemicals should be used. Then use of varieties that are tolerant to diseases should be of our utmost importance, and then timely planting and harvest of the crop are key to success. Now come to the loose smut disease. Loose smut is basically caused by a pathogen used to Labo TTC. It is basically internally seed-borne pathogen, and its incidence is more common in cool and moist northern states. The yield loss reported is up to 2 to 4%, but nowadays the disease has been contained by use of the modern varieties. So you all know that in case of as the symptoms indicate in the photographs, in the infected plant the ears are transformed into black powdery mass consisting of smut spores. The thin covering membrane soon ruptures and the spores are released, leaving the naked rachis. So the teliospores, when you see in the microscope, the teliospores are olivaceous green, spherical to oval and 5 to 9 micron in diameter. They germinate to form germ tube which develops into promycelium with 4 uninucleate cells. In this, sporidia does not form, while in case of common smut or carnal bunt, you can find the sporidia. The infection hyphae is produced from the promycelium after dikaryoticization. Regarding the disease cycle, the fungus perpetuates in the form of dormant mycelium inside the embryo of the infected seed. The mycelium becomes active at the seed germination time and grows along with the seedling in the meristematic tissues. Okay, and it grows but it grows without producing any kind of symptoms. When it reaches the ears, the mycelium invades all the spikelets, and where it grows intercellularly and destroys most of the tissue except the rachis. So this one is the disease cycle, and earlier I told you that it is an internally seed-borne disease, and this disease can be managed by simply treating the seed with Vitavax, which is widely used and very popular in the seed industry. And the Vitavax can be treated at 2.5 gram per kg seeds, or in addition you can use tebuconazole also, which is Raxil at the rate of 1.5 gram per kg seed. These two chemicals are very effective. Now, I have earlier told you that all the modern varieties are nowadays coming which are resistant to the disease. So, you can only find traces of the disease in the field, in the crop. Next disease is foliar blight. You all know the foliar blight pathogen is caused by the two pathogens, that is by Polaris sorokiniana and Alternaria triticina. It was first reported in the year 1924, but in severe form it came during 1960s. And the variety of wheat which were highly resistant to the rust were found to be attacked by this disease. So now you can have the idea that when the crops are 7 to 8 weeks old, the disease start appearing in the field. And the durum varieties of the durum variety of the wheat are most susceptible. Now, regarding the pathogen, it is basically soil as well as seed-borne. The fungus may survive on plant debris lying on soil, provided that the temperature conditions prevailing in the area are favorable for survival of the mycelium, and it is not killed by hot summer. Okay. And the fungus may also be present as conidia as seed surface and mycelium inside the seed coat. So you all know the susceptibility of the plant increases with the age of the plant. So temperature, you can have the favorable conditions like temperature not exceeding 25 degrees centigrade and high humidity. If it is prevailing in the environment, then the disease development is maximum at about 10-week-old seedlings. Now, you also know that it is told that the high sugar and glutamic acid level content in the leaves make them more susceptible to the disease. Regarding the symptoms, the symptom appear as a small, oval, discolorized lesion scattered irregularly on the leaves. The spots gradually enlarge in size and turn brown to grey in colors. Several lesions may coalesce to form large patches. Finally, entire leaves may be killed. Leaves may also begin to dry from the tip. Lower leaves are attacked first, and the symptoms may appear on sheath, ear, awns and gleams. In case of B. sorokiniana attack, the lesions are surrounded by yellow chlorotic halo. So this one is the most distinguishing symptom, that while we having the lesions, if it is surrounded by yellow halo, yellow chlorotic halo, then it is caused by the pathogen Polaris sorokiniana. Now come to the management. So pre-soaking the seed first in cold water for 4 hours, followed by dip in hot water at 52 degrees centigrade for 10 minutes. Serves the purpose of the seed-borne nature of the inoculum, and it has been overcome by that. So seed treatment can be made with Vitavax at 0.2% effective, that effectively controls the disease. Then foliar spray you can have with either dithan M45 at the rate of 0.25% or chlorothalonil, or even if the severity is more, then you can apply tilt. That is propiconazole 25 EC at the rate of 0.1%. And nowadays the varieties are, many varieties have come up which are showing resistance, so better you use the resistant varieties. The third disease is rust. So before coming to rust, you should know that rust is of three kinds. That is black stem rust, which is caused by Puccinia graminestriaceae, then orange rust, leaf rust or brown rust, which is caused by Puccinia recondita, and the yellow rust, also known as stripe rust, which is caused by Puccinia stripiformis. So you can also see the symptoms of all the three rusts, which appear on leaves as well as stem and sheath portions of the plant. Going to rust in detail, you must have the idea about the rust spores. There are 5 kinds of spores, and they are designated. And if a stage O, that is pycnium, so you cannot write stage O for ACM. If you write it ACM, then it will be wrong. So a stage O is designated for pycnium only, then a stage 1 for ACM, and a stage 2 for uridium, and then a stage 3 for tilium, and then a stage 4 for basidia and basidiospores. So in a stage 1, stage 0, pycnium bearing spermacia and receptive hyphae, having the nuclear type is N or N dash. Then ACM, in case of ACM bearing the aciospores, here the hyphae become dikaryotic, having N plus N dash. In uridium also, the hyphae become dikaryotic, remains dikaryotic, that is N plus N dash. While in case of tilium or tiliospore, it becomes 2N, and thereafter meiosis begins in later stage of the tiliospores, and the basidia are formed with N, either N or N dash, not both. So with this, the kind of spore stages. Now you must have the idea about that the rust have some autoecious rust or heteroecious rust, what they are. So the rust which form all spore stages on the same host are called also autoecious. The examples are Velampsora, then Euromyces fabi, while those that form them on two hosts alternating with each other are called heteroecious rust. The most burning example is Puccinia. Okay, now the heteroecious rust, in heteroecious rust, another feature is, in heteroecious rust, the pycnial and acial stages are always formed on one host, while uridial and tilial stages are on the other host. Depending on the spore stages present, the genera and life cycle are designated as macrocyclic when rust fungi producing all the 5 spore stages, or pycnial stage lacking, then it is called macrocyclic. Demicyclic, genera which lack uridial stage, pycnia may be present or absent, and then it is called demicyclic. And microcyclic, genera possess only if tilial stages are there, then it is microcyclic rust. You can differentiate the 3 rusts on the basis of your visual examination also, then microscopic examination. So on visual, you see the part attacked. If it is mostly attacking the stem or sheath portion, then it is a stem rust. Then leaf rust and orange rust mostly attacks the leaves, rarely sheath, very rarely a stalk. In case of brown rust, and yellow rust in leaf is the major part, when severe sheath and a stalk and ear can be affected. Now the time of appearance is for stem rust is March-April. For yellow rust it appears in January, while in brown rust also it appears in January. In case of Pusa condition in Bihar, sometimes it appeared in the late December also. Now In case of, you see the Uridinia, it is large, elongated, running together, and bursting early, throwing large fragments. While in case of yellow rust, it is a small, oval, not running together, arranged in long row. So while in brown rust, it is a small, larger than yellow rust, oval or round, not running together, never in rows. So, and are scattered singly or in small clusters. While in case of Tilia, it is like Iridinia, but black in color. In case of a small rust, burst rather early on all green parts, least on leaf blade, mainly on stems. While in case of yellow rust, generally Iridinia, like Iridinia, but more flattened, dull black, bursts through epidermis, chiefly on lower leaf surface, and all on all green parts. Tilia rarely, you can observe the tilia, but if presents, it is like Uridinia, but more flattened, dull black color, don't burst, and chiefly on lower surface and very rarely elsewhere. So, while in case of Uridinia spores, if it is oval, brown, wall with thick and tiny spines, and its dimension is 25 to 30 into 17 to 20 micron, with 4 equatorial germ pores. While in case of yellow rust, it is spherical to ovate, yellow in color, wall colorless with fine spines, and dimension is 23 to 35 into 20 to 25 micron with 6 to 16 germ pores that are scattered over the spores. Now in case of brown rust, it is spherical, brown to bright orange, wall minutely accumulated, and 16 to 28 micron in diameter, and 7 to 8 germ pores scattered around the surface. Basically rust tiliospores are two-celled in all the cases, and if you see tilia in section, there is no paraphysis in case of a stem rust. Paraphysis divide the tilium into compartment in case of yellow rust, and in case of brown rust, the paraphysis surrounds the tilium. And tiliospore, already I have told you that tiliospore is bicelled, and it is round and pointed at the apex. In case of stem rust, rarely flattened, very thick at the apex, chestnut brown to black in color, and its dimension, you all know, 40 to 46 into 15 to 20 micron in dimension. In case of yellow rust, it is often flattened at the apex, dark brown, less thick at apex, and the dimension is 35 to 63 into 12 to 20 micron. While in case of brown rust, it is often more flattened at the apex, dull black in color, 35 to 63 into 20 to 12 to 20 micron in dimension, and less thick at the apex. Now you can have the idea about the alternate host. So Berberis and Mahonia species and Moho berberis species are the alternate host for black stem rust, while there is no known alternate host for the yellow rust, and Isopyrum, Thalictrum and Clematis species are the alternate host for orange rust or brown rust, you can say. Now collateral host, the Bromus species, Agilops and Hordium are the collateral host for stem rust, while Bromus and Hordium for the yellow rust, and Agilops for the. Now symptoms. Brown rust, black rust, you also call it black stem rust. So and then yellow rust. So black stem rust, elongated reddish brown, that is rusty pustules, most severe on stalks, then on leaf sheath and leaves. Rhodinia merge with each other, soon burst, exposing epidermis, and tilia develops later in the same sorus, darken in color, pustules change from brown to black in later stage. Now in case of brown, orange or leaf rust, Uridinia are confined only to leaves, rarely on leaf sheath and stalks. Earlier I have already discussed. Now it never have in rows, and Uridinia are round to slightly oblong, orange and irregularly scattered or in cluster on the leaf blade. The sori burst early and shed the spores. Tilia may not develop sometimes. In case of yellow rust, it appears earlier than black rust before grains are formed on leaves, and when severe on leaf sheaths and ears also. Uridinia mainly confined to leaves. In each row, a series of oval lemon yellow colored sori are arranged end to end fashion, that is linear fashion. So, dear students, the minuteness of the sori, their arrangement in linear fashion forming stripes, and their lemon yellow color are chief distinguishing macroscopic feature of this rust, that is yellow rust. So with this, the distinguishing symptoms of all the three rust, now we move for the management practices. So excessive use of nitrogenous fertilizers should be avoided, and destruction of alternate collateral hosts and volunteer plants might reduce the inoculum and delay the epiphytotics. So host resistance approaches using multi-line varietal mixture, gene cycle, pyramiding of resistance gene, and using slow rusting or adult plant resistance should be adopted. The varieties like DW88, WH1105, SD2967, DPW621-50 and DBW550 for timely sown conditions, and DBW16, DBW19, DBW71 and PBW590 and WH1021 for late sown conditions have been found resistant to rust. You can Sphaerolopsis phyllum and Verticillium lacani and Gonetobotrytis simplex have been tried as biocontrol agent against rust with limited success. So it has been tried, but the success is very little. So you can manage the disease with the chemicals, and the chemicals like belitone, tilt, that is propiconazole, and plant wax have been found effective in the management of rust. And in case of belitone, if you have to spray, you take 0.1%. Again, tilt, propiconazole, you can also take it 0.1% for effective management of the disease. So the genetic diversity earlier discussed can be generated by multi-line, varietal mixture, mixed cropping, etc., which accord diversity within the field. And on the other hand, gene cycling provides temporal diversity, and a special diversity is achieved by varietal mosaics and gene deployment. So, Karnal bunt. Karnal bunt was first reported from Karnal city by Mitra in the year 1930s, spread to the North India, northern Pakistan, southern Nepal, and parts of Iraq and Mexico. In virulent form, again in 1934 and in 1941, the disease was found in Sindh province and Pakistan, UP and Delhi. The symptoms are, you can find that the grains partially or wholly converted into black powdery mass. The infection spreads along the groups of the grain. The endosperm of the smooth side of the grain is bunted, and partially bunted grain may germinate. The freshly collected grains smell like rotten fish in stinking bunts due to trimethylamine. The pathogen already discussed, that is, it is caused by Neophosia indica. It is basically a floral infecting smut that partially infects the breadwheat, durum, and triticale and rye. Tiliospores are larger and darker than those of field bunt pathogen. The three-layered wall, that is endosporium, episporium, and perisporium of tiliospore, are mixed with subhaline yellowish sterile cells. Disease is not systemic and the infection is airborne. The pathogen survives in soil in form of teliospore or on seed surface. The temperature from February onward were 18 to 22 degrees centigrade and RH above 70% favored the outbreak of the kernal bunt in Punjab. Soil temperature of 17 to 21 degrees C are also conducive for the infection of the crop. So regarding management, the methyl bromide, that is 570 kg per hectare fumigation in wet soil, or VAPAM, that is 1000 liter per hectare fumigation in dry soil, have been found effective. Seed treatment with Vitavax or plant wax at 2 gram per kg seeds have been found effective in experimental conditions. Use of resistant variety like PBW 502, then PDW 233 and WH 956 can be used to avoid the disease. Now the continuous cultivation of the wheat in the same field, deep summer plowing, and avoidance of continuous cultivation of the wheat in the same field should be there. Then follow deep summer plowing, crop rotation with green manure, modifying the sowing dates and amount of fertilizer, number of irrigation and use of clean seeds. By doing so, we can manage the disease. Other diseases, common bunt or smut of wheat. You all know that in the Middle Age, this disease got huge attention, and it helped in the development of the science of plant pathology. So it is also known as the stinging smut or hill bunt. It is in India. It is restricted to cooler regions such as Western UP, parts of Punjab, Himachal Pradesh, and Kashmir. The disease affects the plant by destroying the content of infected kernels and are replacing them with the spores of the fungus. Regarding the pathogen, it is caused by Telesia caris and Telesia foetida. So you know that Telesia caris, that is low smut or rough spored smut, round smut, its teliospore wall has reticulations ranging from minute shallow to meshes to deep indentations of 0.5 to 1.5 micron deep. While in Telesia foetida, it is high smut, also known as smooth spored smut. Teliospore walls are smooth and irregular in shape. Now, the teliospores on germination produce a short, haline, stout promycelium bearing a cluster of 8 to 16 filiform, haline, primary sporidia. These sporidia fuse in pairs in pairs to form characteristic H-shaped structure, and these dikaryotic structures germinate by a hyphae, which then produces secondary sporidia. These secondary sporidia are sickle-shaped. So there are two primary sporidia are H-shaped, while the secondary sporidia are sickle-shaped and haline, and germinate to form the dikaryotic infection hyphae. This hyphae invades the young seedling. So this one is the disease cycle. Now symptoms. The infected plants turn stunted, markedly shorter than normal one, green in color of plant may change to bluish green or grayish green. Diseased plants produce smaller heads. Now pronounced symptoms appear when heads of the plant begin to emerge. Bunt affected plants ripen earlier and ear assume darker green color and are more open than normal one. Infected kernels are shorter, thicker and grayish brown than normal golden yellow or red kernels. Now the smutted grains yield a soft black paste-like mass. In mature grains, black paste-like interiors changes to an oily powder. This is characteristics of the spore mass. All the ears in a smutted plants are attacked, and all the grains in an ear are turned into bunt balls. These are the characteristics. While in kernel bunt, all the grains are not infected, and all the ears are not turned into bunt balls. If the bunt ball is squeezed, it burst open, exposing a greasy, dusty powder composed of teliospores and smelling a strongly rotten fish. Due to volatile compound, earlier told, the volatile compound is trimethylamine. It is the smell of this rotten fish that gives the name stinking smut. So it is also, earlier I told you, the common bunt is also known as stinking smut, and it is the smell of the rotten fish which gives the name stinking smut. You should keep it in mind. Now, management. For management, one should use the resistant varieties, then crop rotation should be followed, the modification of soil temperature and moisture through alteration in date and depth of sowing, destruction of crop debris, help in reducing the soil-borne inoculum. Okay. Now the seed treatment with Beta-Vex at the rate of 0.2% gave the complete control of the disease. Recently, a triazole fungicide, that is beta-tenol, at 90 microgram per kg seeds has been found very effective to eradicate the seed-borne inoculum. So beta-tenol, a triazole fungicide, can be tried in view of the pesticide prohibition by the government. So benzoate should be avoided now, and beta-tenol should be used. Now the last disease is yellow ear rot or tundu disease. So this disease is the characteristic feature of both a fungi, both a nematode and a bacteria. So the nematode associated is Angina TTC. Alone it causes ear cockle disease of wheat, but in association with the bacterium Clavibacter TTC, it causes yellow ear rot, which is also known as tundu disease. So this tundu disease is quite common in India, first reported from Punjab in the year 1917, and a nematode Angina TTC causing ear cockle is found invariably associated with the disease. So earlier the nematode was thought to transmit the bacteria mechanically from galls to the wheat seedling. Later it was confirmed that nematode doesn't play any role in transmission of the pathogen. Okay, so the bacterium is not able to survive as free cells in the soil. The galls formed by the ear cockle nematode are the source of primary inoculum. The disease appears only when the galls of such galls as such from rotting ears are used as inoculum. So you should make it in mind that when the disease appear, the tundu disease will appear only when gall as such from rotting ears are used as inoculum. The bacterium can tolerate sodium chloride concentration up to 3 to 4%, and maximum temperature of 34 for growth is 34 degrees centigrade. So regarding symptoms, the wrinkling and twisting of the basal leaves you can see, then exudation of bright yellow sticky slime enveloping the ear are common. Then the slime binds the leaf sheath, stem and glumes, arresting the growth of the plant and distorting the stem. During wet, the sticky slime trickles down the affected parts, and during dry weather, the slime becomes dry and hard. So these are some of the symptoms of the tundu disease. Now management, seed should be taken from the area which is nematode gall free. Then galls can be removed by floating the seed in brine solution, that is NaCl, at the rate of 40 pound in 25 gallon water. So galls in this soil must be destroyed. Deep summer ploughing helps in eradicating the galls from soil, and disease plants should be uprooted and burned. These are the practices that can be used for management of the disease. So with this, I think your wheat is covered. Any questions you can have, you can again. We will meet again in next lecture. Then we will talk about this. Thank you.